Alright, gentlemen, I wanted to go through Chapter 5 with you one last time to help you review for your quiz. This shouldn't replace studying your chapter, studying your notes, going over stuff that we went over in class, but I just want to hit those high points again so that you can see them. So, if you remember, we started by looking at the lipid bilayer. And so this is an image of that lipid bilayer with the yellow hydrophilic heads and the green hydrophobic tails. And in this particular image, we have a blue protein that's allowing water to go through. And you should remember that this protein is a channel protein known as aquaporin. It's allowing water to move from one side of the membrane to the other. And that becomes important a little bit later in the chapter. So to make things easier on this YouTube video, I put these giant numbers for each section so that if you are skimming through it later, you can find easily the section we're looking at. So we'll first talk about 5.1. 5.1 is basically the section that talks about cellular membranes being a fluid mosaic of lipids and proteins. And remember, we talked about a mosaic being a mixture of all sorts of different things, and so the membrane is made of all combinations of lipids and proteins. And this section really boils down to knowing how to identify the portions that make up a membrane. So again, you've got your phospholipids, your hydrophilic heads, your hydrophobic tails. They form this bilayer with the hydrophobic portions in. The only other sort of lipidy thing that you'll see in the membrane are cholesterols. So cholesterols insert themselves into the membrane because they too are hydrophobic. And we'll talk in a moment about how those can affect the fluidity of the membrane. So you've got this bilayer. Into the bilayer you also have various proteins. So we can have what we call integral proteins. These are proteins that go through the bilayer, meaning that the top and bottom portions of this protein have to be hydrophilic, and the interior portion has to be hydrophobic. There are various roles an integral protein can play, but the basic definition is an integral protein goes through the membrane. And you'll compare that to a peripheral protein, which is found on the side of the lipid bilayer. It can either be on the inside or the outside, and it's peripheral to the entire structure. Additional structures you should note: the cytoplasmic side of the membrane has those microfilaments at the cytoskeleton. So the cytoskeleton comes right up to the inside of the membrane, and it helps support the cell. On the outside of the cell, the extracellular side, you have the extracellular matrix, which we didn't spend a lot of time talking about, but these are made of sugars and carbohydrates, and they provide signaling and recognition on the outside of the cell. These things can link through integral proteins, so you can see the extracellular matrix is linking through these integral proteins to the cytoskeleton. So you don't need to know all the details of these sort of interactions, but just the idea that things can connect through integral membrane proteins. We also have these ideas of glycoproteins and glycolipids, which are just lipids and proteins respectively, that have carbohydrates added to them. Those are often used for cell-to-cell recognition. So one cell can recognize another cell by the composition of carbohydrates that are found on its extracellular side of the membrane. I think by this point you guys are fairly comfortable with the idea that the bilayer has to assemble with the hydrophobic tails in so that they are not exposed to water. So we'll move on from that. The idea that the membrane is fluid comes from these early experiments where if you take a mouse cell with proteins all over it and a human cell with proteins all over it, you fuse those two cells and wait a little bit, you end up seeing those proteins mixed all over the new cell. That suggests that these little proteins that are integral in the membrane are moving all over the place versus staying in one place. And from that we extract the idea that the entire membrane is fluid. It's constantly moving all over the place. Again, this shouldn't be an unfamiliar concept to you because we've watched videos and seen things that really demonstrate that lipid bilayer is fluid. Now, the fluidity of that lipid bilayer is affected by the composition of phospholipids. And the thing is that when we say phospholipid as fatty acid tails plus a phosphate head, there is a lot of diversity in which lipid tails can go onto it. So tails that are more unsaturated are going to prevent packing. just like they do in a vegetable oil. So when they can't pack, the membrane is going to be more fluid. If we have more saturated tails, like we saw in animal fats and in butter, for example, they pack very nicely. When they pack, the membrane can become very viscous and unfluid. One thing that happens for cells is that cells exist in environments with various temperatures, various temperature gradients. They need to be able to manage their cell membrane so it doesn't become either too viscous, in which case nothing will move, or too fluid, in which case things will sort of flow right through it. And one of the tricks that it can do, along with manipulating the composition of saturated and unsaturated fats, is that it adds cholesterol to the membrane. So it's going to reduce fluidity at moderate temperatures. And so it's going to prevent it from getting too fluid because of the interactions between these lipids in the cholesterol and the phospholipid tails. At low temperatures, it's going to prevent it from solidifying because these tails can no longer pack tightly. And so cholesterol kind of serves two purposes there. It keeps things from getting too fluid and also keeps things from getting too viscous. And so that's an important component of the phospholipid bilayer. So again, make sure that for 5.1 you know all of these structures, that you can identify a picture that looks like this, and that you have some concept of what cholesterol is doing in this phospholipid bilayer, and also that this entire thing is fluid and moving all over the place and has lots of things going on. Now when we think about proteins, I want you to think about these integral proteins as going through the membrane. And so this is just showing you an example of one, and we know that this portion above here has to be hydrophilic. This portion below here has to be hydrophobic. And all the stuff in the middle that's exposed to these, the bottom has to be hydrophilic as well. All of this stuff that's exposed to the bilayer has to be hydrophobic, or else it wouldn't be able to exist in this space. These integral membrane proteins can do various things. As we've talked about in class, there's so much diversity in what proteins can do that we attempt to classify them into categories, but just know that all of these categories are sort of loose and there's lots of variability in what's happening in them. But one of the things proteins that go across the membrane can do is help us transport things across the membrane, and we'll come back to that in 5.3 and 5.4. Proteins across the membrane can do enzymatic activity, so basically you can use the membrane to stick an enzyme in a location so that you can actually enhance the ability of that enzyme to do its job because it's not floating around the inside of the cell. We can use it for signal transduction, which is where something on the outside of the cell binds to a protein, causing something to happen on the inside of the cell, basically transducing that signal without actually moving something through the cell. We can use membrane proteins for cell-cell recognition, so these are two different cells. This cell has a glycoprotein on its surface, and this cell has a protein that recognizes that glycoprotein. So now these two cells can basically tell who is who. We can do intracellular joining, and we'll talk about this at the very end when we talk about some of the stuff back from 4.6. Basically, two integral membrane proteins can fuse, causing two cells to also fuse. And as I mentioned on that first figure, we can use integral membrane proteins to attach the cytoskeleton to the extracellular matrix so that we can have connections between structures both inside and outside of the cell. The last idea on the membrane is that if we have all of these proteins sort of going through it, we have all sorts of different compositions of membranes and glycoproteins and glycolipids. Our membrane is sided. So when we have the inside here in blue and the outside here in pink, you should be able to think about the fact that this pink portion stays on the outside as we move through the endomembrane system. And when we get to the end edge of the cell, it's actually going to flip around when it exocytoses, and so the inside portion that's now blue becomes the outside portion of the cell. This can be a complicated idea to think about, but really just think about if you have a bilayer, the two sides don't flip around without any reason. It's not favored for them to change sides, so if they start out by being on one side, they're going to stay on that side until the point comes at the cell membrane where it actually fuses and flips inside out. So in this case, we're flipping inside out. We're not looking at a membrane sort of flipping like that, which would be unfavored. And that brings us to 5.2, which, as I mentioned to you, is perhaps the easiest section in the chapter, and definitely, sorry, perhaps not the easiest section in the textbook, definitely the chapter. 5.2 just says that the membrane structure results in selective permeability. You should have picked all of this up from 5.1, that the cell regulates substance across the boundaries, and these membranes are selectively permeable. They don't let everything through. Those transport proteins are one of the reasons they don't let everything through, because if you are a small nonpolar molecule, you can move through the membrane freely. If you're anything else, you're going to need some help getting through, and these transport proteins will help you get through by making a channel for an ionic or a hydrophilic substance to go across the membrane. And that brings us to 5.3. 5.3 and 5.4, I think, are the two portions of this chapter that you need to pay the most attention to. 5.3 is going to be talking about passive transport. And importantly, you need to remember that passive transport is when things are diffusing across the membrane and there is absolutely no energy being invested in it. So as a preview going forward, passive, no energy; active, which is 5.4, involves energy. That is the basic definition of passive versus active. And so to understand passive, we just need to think about diffusion. So diffusion says that if we have a high concentration of substance here, given enough time, that substance is going to diffuse across the space so that it's equally distributed everywhere. In this particular example, we have a membrane that we have a concentrated set of molecules on one side of the membrane, and eventually when we reach equilibrium, these molecules are going to be equal on both sides, so that we have 1, 2, 3, 4, 5, 6, 7, 8 on both sides of the membrane at equilibrium. That is all we need to think about for diffusion, is that all of the molecules are going to spread out until they're evenly distributed everywhere. If we have multiple solutes, so here we have a yellow solute and a purple solute, they're going to diffuse independently. So yellow is going to seek its equilibrium, purple is going to seek its equilibrium. These guys are going to be moving back and forth across the membrane, but the idea in both cases is that at equilibrium, they are moving back and forth, but the concentrations on either side of the membrane stay equal. Diffusion is really easy, but where it becomes complicated is where we add this next idea of osmosis, which is simply the diffusion of water as opposed to the diffusion of solute. So if we look at this example, we have a low concentration of solute over here and a high concentration of solute over here, and the simple thing you might think about is, okay, the high concentration of solute is going to diffuse to the other side of this membrane so that they equilibrate, except in this example, we're going to put a membrane between them that does not allow the solute, the sugar, to move across. And so to seek equilibrium, to seek an equal concentration on both sides, we're no longer able to move the solute. So what is actually going to move is the water. And so if you think about this, because we have a low concentration of solute over here, we actually have a high concentration of free water. Because we have a high concentration of solute over here, we have a low concentration of free water. So high water is going to move to low water. Water is going to move from left to right, raising the level of this tube on this side. Water has moved this way. At this point, if we were to think about this in terms of molarity, so moles over volume, by decreasing the volume, we've now reached an equal concentration on this side and that side. And that's the basic idea of osmosis. We have some very specific terms that we'll use for osmosis when we think about what's happening in cells. So one of the ways we compare solutions for osmosis by using terms such as hypotonic, isotonic, and hypertonic. When you think about the idea of tonic or tonicity, I want you to think about concentration of solute. Hypotonic means less solute is dissolved. So if we put this cell in a hypotonic solution, the concentration of free water is higher outside than inside, so water is going to move into the cell, causing it to lyse. Water will move in. If we put it in a hypertonic solution, we have more solute outside than inside, so we actually have more free water inside and less free water outside, so water will move out of the cell, causing it to shrivel up. And isotonic means that we have the same amount of solute inside and outside. This would be at equilibrium. The cell would stay the same because the amount of water moving in is going to be equal to the amount of water moving out. This also works in a plant cell, so hypo, iso, and hypertonic. In a hypotonic situation, the cell is going to fill with water just like in an animal cell, but instead of lysing, it's going to actually push against that cell wall, giving this cell turgor pressure, and that's what helps a plant support itself and hold up its stem. If it's in an isotonic solution, it's actually not going to have enough water inside to push against that cell wall, and it's going to wilt. It'll be more flaccid. And if we actually put it into a hypertonic solution, the membranes, as the cells shrivel, will actually separate from the cell wall and will happen what we call plasmolyzed. This cell is now damaged because the membrane has ripped away from the cell wall. So along with osmosis, we can also just think about diffusion of any other substance as being facilitated diffusion. This would be passive transport aided by a protein. Now when we say aided by a protein, we just mean the protein's making a channel. There's still no energy, we've just made a path for those proteins to move. There are two types of transport proteins we can think about. We can think about a channel. A channel is basically making a tunnel that's going to allow molecules to move through. And we can think about a carrier that might add a little more specificity, but it's allowing molecules to go through sort of one at a time or a few at a time, but they're still moving with their concentration gradient in both cases, always going from high concentration to low concentration, which means no energy is being applied. That brings us to 5.4, which is active transport. So unlike everything we just talked about for passive transport, in active transport, energy is necessary. So active transport is going to move things against their concentration gradient, moving them from low concentration to a high concentration, and typically going to use energy in the form of, for example, ATP to get that those molecules to move. The classic example that we're going to talk about for active transport is the sodium-potassium pump. It's one example of this. It's going to move both sodium and potassium against their concentration gradients. So let's look at that. Here's the example of that pump. It's going to be a carrier protein. So what we will look for is that we have a low concentration of sodium inside and a high concentration outside, a low concentration of potassium outside and a high concentration of potassium inside. But in this case, cytoplasmic sodium, which is in the low concentration, is going to bind to the pump at locations that have a high affinity for sodium. This protein will then interact with an ATP molecule, and it'll be phosphorylated. So it'll have a phosphate added to that molecule. That phosphate, as we talked about in class, will change the shape of the molecule, causing it to ultimately flip so that the molecule changes its conformation, putting the sodium onto the outside. And so you can see that here with the sodium on the outside. As the conformation of this protein changes, these locations no longer have a high affinity for sodium, and sodium will float away into the extracellular space. Now that we've changed the conformation, this side of the protein actually has a high affinity for potassium. So the extracellular potassium will be able to bind here, and then this new shape that's now bound with potassium will have the phosphate removed. And when the phosphate removes, the conformation of the protein is going to return to its original shape, putting the potassiums on the inside and allowing those potassiums to flow into the cytoplasmic space because they no longer have an affinity for this molecule. So if we look at this as a cycle, right, we bind sodium, we phosphorylate the protein, that flips the sodium to the outside. We bind potassium, we dephosphorylate the protein, and it flips potassium to the inside. And then we repeat over and over and over again. The goal of this is that we're constantly using ATP to push sodium out into the high concentration and push potassium in where it has a high concentration. Both of these are against the concentration gradient and consequently require energy. So just to look at those ideas, all together, we have two categories of transport, passive and active. Passive is going to require no energy because things are going to go from high to low. Active is going to require energy because they're going to go from low, if you look at the circles, to high, if you look at the circles up here. In passive transport, we have both diffusion and facilitated diffusion. We'll use the term diffusion when we're talking about things that can move right through the membrane, small nonpolar molecules such as oxygen or carbon dioxide. Facilitated diffusion is going to be anything that requires a protein to get through. That would include water, any sorts of hydrophilic or ionic molecules. Active transport is requiring energy so that it can move things against its concentration gradient. And so hopefully this diagram helps you see the two distinctions between these types of transport. Osmosis would be an example of facilitated diffusion using one of these aquaporin channels, and that would just be the specific diffusion of water. So when we think about these active transports, one of the things we can do is pump ions and create a membrane potential. So membrane potential is voltage across the membrane, and if we create a voltage difference by pumping ions to either side of the membrane, we can actually use that voltage to do cellular work. And we'll call this an electrochemical gradient because we've moved chemicals from one side to the other, creating a concentration gradient, but because those chemicals are also charged, we're going to change the electrical force, the membrane potential, based on moving those ions. An electrogenic pump is something like the sodium-potassium pump that lets you do this. But to make things simpler for you guys, we talked more about a proton pump in class. So unlike the sodium-potassium that's moving ions in both ways, when we think about a proton pump, we're just going to use ATP energy to move a proton from one side of the membrane to the other side of the membrane. And by doing that, we're moving positive charge to one place, and we're creating a positive charge on one side of the membrane and a more negative charge on the other side of the membrane. And so, some of you wondered, well, that's really cool, but why would you put all those protons in another place? Why would you create this membrane potential? And the reason is that you can use this membrane potential to do cotransport, which is coupling the active transport of something with the passive transport of something else. And so in this example, we consider the proton pump that's moving hydrogens to one side, creating a membrane potential, more positive out here, more negative out here. And because we're doing that, we have another transport protein that's going to highly favor the movement of H plus in to the cell, both because the concentration of H plus is high out here and low in here. So we've got a chemical gradient, high to low, and we also have an electrical gradient from positive to negative. These positive charges are going to be attracted to come in, but we don't just let them come back into the cell. We force them to go through a cotransporter that brings a sucrose molecule with it when a hydrogen comes in. And so now we've coupled the movement of hydrogen passively across the membrane with an active transport against the concentration gradient of sucrose. So by doing work all the way down here, we're creating a membrane potential that can do work later to move something like sucrose into the cell. This proton pump might be putting lots of protons on one side of the membrane, and then multiple cotransporters are moving lots of different things into the cell, which is why you're going to separate these two jobs spatially within the cell. Those are the two big ideas, and that leaves us with just 5.5 and 5.5, 4.6 and 5.6 to sort of gloss over to make sure you know some terms. 5.5 talks about bulk transport. Bulk transport is just the idea that if you're considerably bigger than a channel protein or a carrier protein could be, we can't use those proteins to move you across the membrane. So large molecules like polysaccharides, proteins go in bulk by using vesicles. This is also requiring energy, so it's also active transport. Phagocytosis is where the cell just wraps its membrane around a food or another particle and pulls it into a vacuole. And so it basically just wraps around it, makes a vesicle, pulls it into the cell. And you guys know from the previous chapter that this vacuole will then fuse with the lysosome that can break down all of the stuff within the vacuole. So if the cell pulls in a large solid outside of the cell, that's going to be phagocytosis. You can compare that to penocytosis, which is where the cell just pulls in liquid from outside of the cell and all of the proteins that are dissolved within that liquid. So this doesn't have much specificity. It's basically just grabbing a mouthful of liquid, and whatever's in there is what's in there, and the cell will then use that for its needs. The cell can add a little more specificity to this using what we call receptor-mediated endocytosis. And so where this would be like fishing with a net, where you're just going to cast the net and catch whatever you can, Receptor-mediated endocytosis uses receptors in a way that you could think about bait on a net. So these receptors bind a protein of interest, and then they are brought into a vesicle. So now you've highly enriched for the molecules that you want at these receptors. So that gives you three ways to move bulk contents into the cell, and just know those three terms. So phagocytosis is going to move in full particles, pino is going to move in liquid, and receptor-mediated endocytosis is going to give you some more specificity to actually pull in dissolved particles that the cell desires to have inside of it. So 4.7 is our throwback from the previous chapter, and that's sort of saved for this unit because I think that you guys will have a better time understanding what was going on in 4.7 now that you know a little bit about the membrane. Because 4.7 is talking about the extracellular components and connections that help coordinate cellular activity. So this is really talking about the stuff that's happening at the membrane to help communication and other collaboration between adjacent cells. So one of the things we talked about was the cell wall. Hopefully now the cell wall makes a little more sense to you in terms of its ability to prevent excessive uptake of water. So when we talked about osmosis in a plant cell, we said that the cell can't actually take up too much water and lyse because that cell wall is going to give it structure and allow it to take up extra water and be what we call turgid. These plant cell walls are made of cellulose, so they're made of carbohydrates. And while I'm not overly concerned about you knowing the terms, we can have a primary cell wall and a secondary cell wall, and then a lamella, which is space between adjacent cell walls. And you can see the primary cell wall is on the outside of the cell. The secondary cell wall is right inside of that primary cell wall in some cells, so between the primary cell wall and the cell membrane. And the lamella is just going to be the space in between two adjacent cells. An important idea that I want you to think about, and we'll come back to it in a second, is the channels that you're seeing between these adjacent cells, and we call those the plasmodesmata. So those are channels between plant cells that will actually allow fluids and sugars and some small proteins to flow between adjacent cytoplasms. And so in a multicellular organism, this becomes a way that materials can be shared between the cells in a much easier fashion than excreting it to the exterior environment and then importing it back in. In animal cells, instead of having a cell wall, we have what we call an extracellular matrix. This is made up of various glycoproteins, so combinations of carbohydrates and proteins. Collagen, proteoglycans, fibronectin are among these. The important thing you need to know is this just exists outside of the cell. It can provide some layer of cell recognition, some layer of cell structure, cell protection, and because you can see it here on this picture of the bilayer, know that it can interact with integral membrane proteins and consequently the cytoplasm and materials in the cytoplasm such that you can have interactions between the two, between the cytoskeleton, the integral membrane proteins, and the extracellular matrix. That's about all you need to know for the extracellular matrix. Finally, I wanted you guys to think a little bit about cell junctions. And so cell junctions are places where the cell can actually cause linkages between adjacent cells sharing proteins that go through both membranes. We already mentioned the plasmodesmata. These are channels that perforate the plant cell walls, and water and small solutes can pass from cell to cell. And then in animal cells, we have three main types. We call tight junctions, desmosomes, and gap junctions. All of these are especially common in epithelial tissue, so tissue that makes up the outer layers of skin. And so you can see here, these are epithelial cells. Tight junctions actually link cells together really tightly, preventing fluid from moving across these layers of cells. So this, for example, is why when you stick your hand in water, all of the water doesn't flow through to the inside of your body. These tight junctions sort of hold things together. Desmosomes are another structure that's going to hold cells together, prevent them from shearing apart, moving past one another. You can think about them as rivets that are sort of holding adjacent cells together, and they're using intermediate filaments as the cytoskeletal structures to provide that stability. And finally, you can have what's called gap junctions, which are sort of like channel proteins that move between two cell membranes. And so now we have a direct channel for ions or small molecules to move from this cell into that cell. And so these are just sort of more complex examples of membrane proteins and the types of interactions they can have with cells that are near them. And that finally brings us to 5.6, which is cell signaling. And again, the only thing we want to think about for 5.6 is just this idea that now that we know about the membrane, stuff is going to happen at the membrane. So cells are going to interact at the membrane. And the site of all cellular signaling is going to start at the membrane because that's the outermost limit of a cell. And so cell-to-cell communication is going to be important for both multi and unicellular organisms. And all you need to know right now about communication is that there are basically two types. There is local signaling. This would be where a single cell secretes some sort of molecule that tells the cells around it to do stuff. We would call that paracrine signaling. A very specific example of local signaling actually occurs in your neurons, and we call that synaptic signaling. So this would be a neuron signaling the firing of another neuron by releasing neurotransmitters across the synapse. These are ideas that you're probably somewhat familiar with, but again, you don't need to know much about it other than that a local release of chemical is causing a localized response in a cell next to it. So we'll compare paracrine signaling to endocrine signaling, and endocrine signaling is going to be when we have a long distance signal. And so in this case, instead of a cell that's right near another cell releasing some sort of chemical, this cell is going to release some sort of molecule that's going to flow in humans, for example, throughout the bloodstream. And it's going to cause lots of responses in lots of places throughout the body. And so what we're really thinking about is just a difference in scale here, that one cell is actually releasing molecules to a lot of different cells, causing a lot of different responses. So the only other idea I'd like you guys to know about cell signaling is just a basic concept of how a signal might work. And that's basically looking at three steps of cell signaling, which we would call reception, transduction, and response. This sounds more complicated than it actually is, so I'll just show you the figure. The idea here is that when a cell receives a signal, it's going to receive it at the membrane. So we have an integral membrane protein that's going to receive the signal. That received signal, reception of that signal, is going to trigger a bunch of different chemical reactions that's going to relay that signal throughout the cell. We'll call that transduction. So reception, transduction, once you've transduced the signal to where it needs to go, that would be the response. So the three steps are reception, transduction, and response. And we can see that again in another example here, right? Reception, some growth factor is hitting a receptor. Transduction, lots of different interactions, enzymes, molecules are amplifying the signal throughout the cell. And ultimately we get a response where something happens, for example, at the DNA to cause the cell to react to what's going on. That should hopefully be a pretty good run through of everything that you need to know for your quiz. Make sure, again, you've reviewed your notes, you've reviewed the material we've gone over in class. I want you to think a lot about the lab we did, even though you didn't write that up. Make sure that you think about what you did in that lab and the types of results you saw in that lab. And also, I highly recommend that you take advantage of the online practice test questions and quizzes so that you can be ready for our quiz on Chapter 5. Good luck. Send me an email if you have any questions.